Good morning. In this video, we will be discussing about glycogen metabolism. This metabolism of glycogen, I will be discussing under the following headings. To start with, a brief introduction about the structure and functions of glycogen. Next, site, that means where the pathway takes place. Next, how the pathway takes place. Then, how this pathway is regulated, and finally, the clinical significance related to the pathway. So these are the headings under which I will be discussing about glycogen metabolism. In this video, I will restrict to the first three headings, that means introduction, site and pathway, whereas regulation and clinical significance I will be discussing separately in the next video. Okay, so first we'll start with the introduction part. Glycogen is actually a branched homopolysaccharide made up of glucose residues which are connected by alpha 1,4 linkage, except at the branch point where it is alpha 1,6 linkage. This is the structure of glycogen. The main function of this glycogen is it acts as a storage form of glucose and it is stored mainly in liver and muscle. So this stored glycogen, whenever there is a need, that means whenever during starvation or fasting, it will be converted back to glucose, especially the liver glycogen, it will be converted back to the glucose and it plays an important role in maintaining the blood glucose level during the early fasting time. So this is the main function of glycogen. Okay, so this is a brief introduction about the structure and function of glycogen. Next, we will move on to the site. Just now I told you that glycogen is stored mainly in liver and muscle. So the pathway of glycogen metabolism takes place mainly in liver and muscle. Within this liver and muscle cells, it takes place exclusively within the cytoplasm. So most of the enzymes involved in the glycogen synthesis or glycogen breakdown, they are present in the cytoplasm. So the pathway takes place mainly in the cytoplasm. Okay, so inside the cytoplasm, so how the pathway takes? So what are the reactions are involved? We'll be seeing next. Okay, so under pathway, we'll be discussing both about the reactions of glycogen synthesis and also glycogen breakdown. First, we will start with glycogen synthesis. The pathway of glycogen synthesis can be broadly classified into four phases. So what are those four phases means? The first one is activation of glucose. Next, initiating the process, elongation, and finally branching. So these are the four major phases of glycogen synthesis. First, we will start with activation of glucose. So in this phase, glucose will be first converted to its active form, which is actually involved in the glycogen synthesis. So that active form, which actually directly participates in the glycogen synthesis, is nothing but UDP glucose. So in this first phase, under the activation of glucose, we will be discussing how glucose is converted to UDP glucose. So how it is converted means, first in any glucose utilization pathway, the first step is phosphorylation of the glucose at the 6th carbon to form glucose 6-phosphate. The enzyme for this reaction is hexokinase or glucokinase. In the liver, it will be catalyzed by glucokinase, whereas in the other tissues, especially in muscle cells, it will be done by the hexokinase. Okay, so now this glucose 6-phosphate form will be converted to glucose 1-phosphate by an enzyme called as phosphoglucomutase. Okay, so mutase is a type of an isomerase enzyme which will, as the name says, mutase, it will be silently shifting the functional group or any group. Here it shifts the phosphate group from one carbon to other carbon atom. Okay, so that is a function of mutase. So the same mutase enzyme will convert glucose 6-phosphate to glucose 1-phosphate. Now this glucose 1-phosphate in turn will react with UTP. Okay, so during this reaction, what will happen means 1 phosphate from glucose 1-phosphate and 1 phosphate from UTP will be removed as pyrophosphate. Okay, so when you remove the pyrophosphate, so when you remove 1 phosphate from this glucose 1-phosphate, it will be converted to glucose. Similarly, when you remove 1 phosphate from UTP, triphosphate, it will be converted to UDP, diphosphate. Okay, so now this glucose and UDP will join together to form UDP glucose. Okay, so this is how UDP glucose or the activation of glucose takes place. Okay, so for this reaction, the last reaction, it is catalyzed by the enzyme UDP glucose pyrophosphorylase. Okay, since pyrophosphate is removed to produce UDP glucose, this enzyme is named as UDP glucose pyrophosphorylase. Okay, so it is one of the main enzyme which is needed for the activation of glucose. That means for the synthesis of UDP glucose. Okay, so now with the help of this UDP glucose, how the process is initiated, how it is elongated, we will be discussing in the subsequent slides. First, we will start with the process of initiation. Okay, so how the glycogen synthesis is initiated. For initiating the glycogen synthesis, we need a primer called as glycogenin. This glycogenin is actually a dimeric glycoprotein. That dimeric means it is made up of two units. Glycoprotein means it is made up of, it along with protein, it also contains small few residues of glucose molecules. Okay, usually 4 to 6 glucose residues will be attached to each of these monomers of the glycogenin dimer. Okay, so now to the already existing glucose residues, the first glucose residue will be attached. Okay, and this process is called as initiation of glycogen synthesis. Okay, so by how this first glucose residue is added means by this chemical reaction. Okay, so the already glycogen primer N is the number of glucose residues which are already existing. Okay, so to that UDP glucose will transfer the glucose so that glycogen becomes N plus 1. The glucose residues will be increased by 1 and with UDP will be released, it will be sent thrown out from this thing. Okay, so this is how the initiation of glycogen synthesis starts. Okay, so initiation is nothing but addition of first glucose residue to the already existing glucose residues of glycogenin primer. Okay, so this process is called as initiation of glycogen synthesis. After initiation, that means after the addition of first glucose residue, further glucose residues will be added in the next phase called as elongation phase. Okay, so here what will happen means, so in the same way, by the same reaction, the glucose residues will be added one by one. After the first glucose, then subsequent glucose will be added one by one and it will be attached in the alpha 1,4 linkage. Okay, so by the same reaction, the glucose residues will be attached in the alpha 1,4 linkages. Okay, so since both initiation and elongation follows the same pattern of reaction, the enzyme for these two processes also is same. So both initiation and elongation of glycogen synthesis is done by an enzyme called as glycogen synthase. Okay, which is one of the major enzyme and the rate limiting enzyme in glycogen synthesis process. Okay, so that's it about the elongation. So initiation over and elongation over. Okay, so how long this elongation will continue means this glycogen synthase will elongate the chain length up to 11 or 12 residues. After some 11 or 12 residues of glucose are attached, what will happen means it will move on to the next phase called as branching phase. So here what will happen means the glycogen synthase, after it has elongated the glucose residues to 11 or 12 residues, what will happen means next the branching enzyme will come into act. So what is the action of this branching enzyme means they will detach the last 6 residues from the linear chain. They will be detaching it and they will be attaching it as a new branch through alpha 1,6 linkage. Okay, so this is called as branching. This process is called as branching and this is done by an enzyme called as branching enzyme or otherwise called as amylo alpha 1,4 to alpha 1,6 transglucosidase. Okay, so the transglucosidase means, as the name says, it transfers the glucose residues, the last 6 residues from alpha 1,4 linkage. Actually, normally in the linear structure, it is attached in alpha 1,4 linkage. Okay, so that is detached and it is attached as a alpha 1,6 linkage. Okay, that is why this enzyme is called as branching enzyme or amylo alpha 1,4 to alpha 1,6 transglucosidase. Okay, so now with the help of this branching enzyme, we have got two different branches. Now these two branches again will be elongated by our glycogen synthase enzyme. Okay, so how long it will be elongated? Same way, up to 11 or 12 residues it will be elongated. Okay, after that the branching enzyme will come into the action. So this branching enzyme, again what they will do, the last 6 residues it will be detaching it and it will be attaching at as a new branch at alpha 1,6 linkage. So again these two branches will be elongated. Okay, so the repeated cycles of elongation by glycogen synthase and branching by branching enzyme will repeat many times and it will result in the synthesis of highly branched glycogen molecule. This is how glycogen is synthesized in our body. Okay, so to sum up, the glycogen of glycogen synthesis is made up of 4 phases: activation of glucose, initiation, elongation, and branching. Okay, so first activation. What is the activation process? The conversion of glucose to UDP glucose is called as activation process. Okay, so for this activation we need an important enzyme called as UDP glucose pyrophosphorylase. Okay, so after activation, so initiating the process we need a primer, a glycoprotein called as glycogenin. Okay, so this glycogenin will be having some 5 to 6 residues of glucose already attached to it. To that already existing glucose residues, the first glucose residue will be added, that is called as initiation. After the first residue is added, further glucose residues will be added in the similar fashion in alpha 1,4 linkage, that is called as elongation. Okay, so both this initiation and elongation is catalyzed by the enzyme glycogen synthase. Okay, so this glycogen synthase, it usually elongates the residues up to 11 or 12 in size. Okay, so once that 11 or 12 is achieved, what will happen means the last 6 residues will be detached and will be attached as a branch by the branching enzyme, which is otherwise called as amylo alpha 1,4 alpha 1,6 transglucosidase. Now to the two new branches, further glycogen synthase will be elongating it. Again up to 11 to 12 residues it will be elongating, then again branching, again elongating. So this repeated cycle results in the synthesis of highly branched glycogen molecule. Okay, so this is how glycogen is synthesized in our body. Next, coming to glycogen breakdown. So just now we discussed that glycogen synthesis is nothing but a process which involves a repeated action of elongation and branching. So elongation and branching. So this repeated cycle results in glycogen synthesis. Okay, so for glycogen breakdown, it is just the opposite. Okay, so what is the opposite for elongation? It is shortening or trimming. Opposite for branching is debranching. So the repeated cycles of trimming and debranching will result in the process called as glycogen breakdown. Okay, so now we will see in detail how each of these reactions takes place. First, we will start with trimming reaction. Okay, so consider this is one of the open end of the glycogen molecule. So now from this non-reducing end, the glucose molecules will be removed one by one by the enzyme, by the trimming enzyme, which is otherwise called as glycogen phosphorylase. Okay, so glycogen phosphorylase, and this enzyme needs pyridoxal phosphate, vitamin B6 acids coenzyme. Okay, so it is an important MCQ thing. The only enzyme in the carbohydrate metabolism which uses B6, vitamin B6 or PLPS, this glycogen phosphorylase. Okay, so what is the action of this glycogen phosphorylase means? As the name says, it uses a phosphate molecule and it breaks the glucose residues from the non-reducing end. Okay, so the glucose molecule will be released as glucose 1-phosphate every time. So every time one by one the glucose molecule will be removed as glucose 1-phosphate. This action of glycogen phosphorylase will continue till it reaches 3 to 4 residues away from the branching point. Okay, so the glycogen phosphorylase can continue its action. It can continue only up to 4 residues away from the branching point. Beyond this, the glycogen phosphorylase cannot remove the glucose residues. Okay, so at that point, the next enzyme, debranching enzyme, will come into action. So this debranching enzyme, actually it is made up of 2 components. It has 2 enzyme activity. The first component is glucan transferase and the second component is alpha 1,6 glucosidase. Now, what is the action of these 2 components means? Initially, the glucan transferase enzyme, as the name says, it transfers the glucose residues. Which glucose residues means? The last 3 residues from the branch point will be transferred to the other branch. Okay, so that what will happen means the glucose residue which is attached with alpha 1,6 linkage will be exposed. Okay, so the glucan transferase will transfer the glucose residues from the last 3 residues from one branch to other branch, and it exposes the alpha 1,6 glycosidic bond. Now, to this exposed alpha 1,6 glycosidic bond, this enzyme alpha 1,6 glucosidase will break this bond and it will release the glucose molecule so that the branched point will be converted into a linear structure. Okay, so please note here, when the glycogen phosphorylase is removing the glucose residues, it will be removed only as glucose 1-phosphate. But when alpha 1,6-glucosidase is removed, it will be removing as a glucose. Okay, so it mainly we get glucose 1-phosphate, but along with that we also get glucose. Usually they will be in the ratio of 8:1. Glucose, for every 8 glucose or 1-phosphate molecules released, 1 glucose will be released from the branching point. Okay, so this is how the branch structure will be converted to a linear structure. So once the linear structure is formed, again the trimming enzyme, the glycogen phosphorylase, will come into action. So they will be removing the glucose residues one by one from this corner. Okay, from this non-reducing end, it will be removing it. Okay, so how long it will remove? Again, same thing, till it reaches 4 residues away from the branching point. Okay, so once that point comes, then again debranching enzyme will come and they will convert the branch point into a linear structure. Again trimming enzyme will come. Okay, so this repeated action of trimming enzyme and debranching enzyme will result in the production of large molecules of glucose 1-phosphate molecules. We will be getting large quantities of glucose 1-phosphate molecules. Okay, so what is the fate of this glucose 1-phosphate means? Initially, it will be converted to glucose 6-phosphate by the same phosphoglucomutase enzyme. The mutase enzyme, by the reversible reaction, it will be converting glucose 1-phosphate to glucose 6-phosphate. Now, this glucose 6-phosphate, by the action of the enzyme glucose 6-phosphatase, as the name says, it will chop the phosphate, it will remove the phosphate and converts it into glucose. Okay, so this is how glycogen breakdown takes place and glycogen is converted into glucose molecules. Got it? Now, one important thing is this enzyme glucose 6-phosphatase is present only in the liver. Okay, which means only the liver glycogen can be converted to glucose and it can be used for glucose homeostasis, for maintaining the blood glucose level. Okay, whereas in muscle, what will happen means the glucose 6-phosphate cannot be converted to glucose. So instead, what it can be done means it can be oxidized by the muscle cell to form by anaerobic glycolysis, and it will be used releasing energy which will be utilized for its own muscle contraction. Okay, so that is the fate of the glycogen stored in muscle. Okay, the glycogen stored in liver can be used for maintaining the glucose homeostasis, whereas the muscle glycogen cannot be used for that purpose. Okay, so this is how it takes place. So in the muscle cell, when the glucose 6-phosphate undergoes anaerobic glycolysis, normally when glucose undergoes anaerobic glycolysis, we will be getting 2 ATPs. When glucose is converted to lactate, we will be usually getting 2 ATPs for each glucose molecule. But here what is the thing is, when you take directly from glycogen, we are directly getting glucose 6-phosphate. Okay, so what is the thing? Normally in glycolysis, the first step, we will be adding one phosphate, or I will be adding one high energy phosphate. We will be investing one high energy phosphate to form glucose 6-phosphate. Here, since we are directly getting glucose 6-phosphate from glycogen, that energy investment step is bypassed. Okay, so 1 ATP is conserved. So totally we get 3 ATPs when glycogen, when one glucose molecule from glycogen is oxidized under anaerobic glycolysis within the muscle cell. So normally in other cell, when you start from glucose, you will be getting 2 ATPs in anaerobic glycolysis. But in the muscle cell, when it starts from glycogen, we will be getting 3 ATPs from each of the glucose residues. Okay, so that's it about the glycogen breakdown and glycogen synthesis. So with this, I'll stop this video. So the regulation part and the clinical significance I'll be discussing in the subsequent video. Thank you.